So hello everyone, my name is Freya. I'm from the same group as Olivero and Arseni, and today I'll be talking to you a little bit about what I am doing during my postdoc, and I'm focusing pretty much on hormones. So with the hope that this works for me, cool. So you might all be familiar with the endocrine system. So we have in our bodies things called hormones, which are chemicals that pretty much communicate throughout the whole body, keeping a certain level of homeostasis. And you might be familiar with things like adrenaline with the fight or flight response, or even cortisol and insulin. So when you're taking food in, either insulin is there to store the food that you can use it later, or cortisol really is there that when you have a moment of stress, you're actually using up the nutrients. But then, of course, we have sexual hormones like testosterone that is typically higher in males, and estrogens that are typically higher in females. And then we have, for example, what we would know in us humans as oxytocin, but in our cool teleos fish, we have it as isotocin. And this is what we call the cuddle hormone, or it's very important fish for mating and pairing. And then similarly, progesterone is really important also for development and fertility. And last but not least, prolactin is the parental care hormone. So this in humans, for example, is really important with lactation and that the mothers are not killing the offspring immediately. But this also in fish, we see it as potentially fanning the eggs or protecting the eggs from predators. So why is this all important? Well, we are working with fish, and unfortunately, we have so far a lot of knowledge when it comes to mammalian pregnancy. So we know throughout mammalian pregnancy all the different hormones and how they fluctuate, but there's not a lot of information actually on male pregnancy. So I won't go in all the detail because Arseni has just done that, but we have a male pregnancy gradient, voila. And so for us, it's interesting to know, okay, how do these hormones actually work with male pregnancy? Like, is there complete reversal or things conserved or not? The other things that are really cool to say in the fish that we work with is that actually we can separate something that would be sex, so sperm or eggs, and sex roles, so what we consider as male pregnancy. So these two things are uncoupled. Furthermore, also really cool, we have different mating systems, different degrees of sexual dimorphism, from some species having much stronger sexual dimorphism, for example, like Neurophis, where the females are really colorful on the side, and they compete for access to the males, to other cases where there's maybe less sexual dimorphism. And really important that I have to mention is that in our cygnatids, we still find, despite the male pregnancy, that the females are producing a lot more estrogen and the males are producing a lot more testosterone. So there seems to be something that is conserved in that respect. Now, getting to a little bit of what my research questions are. So the first thing we really wanted to look at, because some of the research and literature is a little bit conflicting, was to look at, if we look at different cygnatid species along the male pregnancy gradient, so from seahorses to Neurophis, how does these different hormones actually change, and what are their concentrations? And on top of that, I'll come to it, I've also added Hydrangea formosa, which is a fish species with female pregnancy, so we have a comparison. And then the second question really was looking, okay, not just at the baseline, like at the top of the different hormones between these different species, but actually when we look at two species with one at a higher degree of male pregnancy, so Cygnatus typhli, and one with just the eggs attachment, like Neurophis ophidian, how do the different hormones change across the male pregnancy? So do we see things that are similar like mammalian pregnancy or not? And as I was saying before, I did these two experiments looking at 7 different hormones, just to kind of quickly go through with you again. So we have testosterone, 11-ketotestosterone. 11-ketotestosterone is only found in fish. It is a derivative of testosterone, but it is really important in fish. It does mostly of the male androgen functions. Then, of course, cortisol, as mentioned before, estradiol also, progesterone, prolactin, and isotocin, which I said is the fish-specific oxytocin. So these are the hormones that I was looking in all these different species. So just to give you a beginning of what I did, so as I said, we have Hippocampus abdominalis, Cygnatus typhli, Neurophis ophidian, and Hydrangea formosa. Hydrangea formosa has a high degree of female matrotrophy index, so they are also viviparous species. And we took males and females. We then took different tissues. For some individuals and species, it was easier to take just the liver because the testes are sometimes so small you can't really extract hormones from it. And then we did so-called ELISA immunoassays, which is pretty much you put your hormone in, you have a few antibodies and enzymes, things fluoresce, and you can actually measure what's the hormonal concentration. And then last but not least, we normalized everything to be able to compare it. Now, coming to the results. So, just to explain, this is a heat map. So what we're looking at is pretty much all the hormones on this axis, and then we can look at the different species aggregating. So we have the different colors here, and then we have also the separation with the different species and the tissues, but I'll get to each thing individually. So this is just a way to kind of aggregate the data so we can really look at the different comparisons. But important is, so we had a real strong significant difference when it came to the different hormones for species and tissue. But most importantly, which was kind of expected, the strongest difference was really between Hyperandia and all the different cygnatids. So there clearly is a real strong separation when it comes to the overall hormonal picture with a species that has female pregnancy and those that have male pregnancy. So this was the first core finding. But then also we can see there's a different gradient when it comes to similarities between the different cygnatids. So to that point, I kind of wanted to focus here with you today just on the pouch structure, because this is the male pregnancy structure. So the main thing that we can see that Neurophysophedium pouch has just so much high concentration compared to Hippocampus abdominalis, for example, and Signatus tifla pouch. So this is in of itself really interesting that we could find that Neurophysophedium had the strongest difference, significance difference, between Signatus tifla and Hippocampus abdominalis. But when we're actually looking at Signatus tifla and Hippocampus abdominalis, there was not such a strong significant difference, which is an indication potentially that actually, because they have this brooding structure, they have to have maybe potentially more similar hormonal profiles when it comes to pregnancy. On top of that, when we are actually looking at sexual dimorphism, which I mentioned before, across the different species, what we can see when we're comparing, for example, Neurophysophedium liver male to liver female, because we didn't have the testes for this species, we can actually see that even here, the liver male was really high with of different concentrations of hormones, but not in the case for the female specifically. And so there was a strong dimorphism also here. So it's not just what we see physiologically and for colors, but it is also hormonal. And Signatus tifla, this was not the case with all the hormones, but it was the case with sex-specific hormones, which in and of itself is really interesting. So we can see that for testosterone, 11-ketotestosterone, it tends to be high in the males, and estradiol tends to be high in the females. So at least we know that some particularly sexual things are conserved. But then when we go all the way to, again, the seahorses, we find that when it comes particularly to testosterone and 11-ketotestosterone, there doesn't seem to be much of a sexual dimorphism, so it seems to be that they're a little bit more similar. In the estradiol, it was also slightly significantly different when we were looking at ovary and testes, but the main interesting thing was we could see that in hippocampus, there does seem to be a lot of prolactin being produced, and this was not the case in the testes. So being a species with really high degree of parental care, this might be something really worth looking into. Okay, and now we come to the second experiment, and here I really wanted to look at, I said, throughout the male pregnancy, how things look like. So to do that, we got the pouch of Signatus tifla throughout the pregnancy, and also of Neurophysophedium. It's not a proper pouch structure, but it's pretty much the skin tissue where the eggs would be attached. And what we did for both species, we mated them, and then after 3 days, so that was early pregnancy, we took them. After 10 days, which was mid-pregnancy, we dissected them, and then at late, that was 20 days, and then at birth. So the moment they gave birth, we also dissected them. And also here, we did the 7 different hormonal analyses with the ELISAs. Now, this is a PCA. It's just pretty much a way to represent, again, all the data that I had of all the different hormones together. So you have each single dot is an individual, and each axis represents what the variation is trying to explain. So for example, what we see here is that actually the strongest difference was the tissue. So we have the Signatus tifla liver and Neurophysophedium liver down here, and then here we have the pouch of both species. So there seems to be that in of itself, the two different tissues are really dimorphic. But the cool thing is that we see a really strong, significant difference between species and stage of pregnancy, which is the next thing I'm going to come to. So what you can pretty much see here is that we have the control, which is non-pregnant individuals. We go to early pregnancy, mid-pregnancy, late, and birth. And I'm going through all the different hormones and how they fluctuate throughout pregnancy. So what we see is that there seems to be an initial drop at the beginning of early, but then it goes fairly back up. And this is for estradiol and prolactin, both hormones really important in pregnancy. And progesterone, for example, that stayed fairly consistent and went lower at the end, and cortisol had a little spike. This is somewhat similar when we think of female pregnancy. So we can see estradiol kind of going up, and prolactin too, and cortisol also peaking at the end. For progesterone, it doesn't seem to be the case, however. So there do seem to be some similarities when we're still thinking of female pregnancy with male pregnancy hormonally, but not entirely. However, when we're looking at a species that doesn't really have proper pregnancy, we can see there's this massive drop of hormones. So it seems that they probably need these hormones when it comes to like courting or engaging, but not necessarily throughout pregnancy. And this, as I said before, is endocrine, as you all know, is just attaching the eggs. Now, when we look at the other hormones, so 11-ketotestosterone, testosterone, and oxytocin, what we can see is actually interesting. Testosterone stays fairly low throughout pregnancy. This also matches what we see in humans. Oxytocin, interestingly, spikes at the beginning, which could have something to do with actually maybe potentially the embryos attaching inside and that being useful. But the interesting thing was that 11-ketotestosterone actually, despite some variability, tends to go up. This is a little bit unexpected in on itself because you would think that ketotestosterone might be detrimental actually to the pregnancy itself. But that doesn't seem to be the case. And we know from other literature that actually 11-ketotestosterone can be useful when they're developing as young individuals to create this pouch. But it seems that this might also be really useful during pregnancy. And again, nidophosophidian seems to be a little bit the exception. Everything goes down, but things here were a little bit more stable.
3 minutes left.
Cool, I'm getting towards the end. I'm perfect. So, pretty much, to just give you a summary. So these are all the hormones we looked at, and what we can see that it is important that we can see these hormones. There is a sexual dimorphic hormonal profile, strongest when we look between nidophosophidian and hippocampus abdominalis, which kind of matches what we know. And there is a hormonal shift throughout pregnancy, and to some degree, some things are more similar when it comes to the mammalian pregnancy, and other things are maybe novel and less conserved with the 11-ketotestosterone. But now, why is this all important? Well, first and foremost, we have to look at the big environmental picture. So we can really see that actually we have a lot of chemicals, and for example, the baby pill that produces a lot of estrogen going in the water systems. We have sunscreen and so on, and these are all endocrine disruptors and can actually be very detrimental to the fish. We can also see that, for example, there was a study done in the Baltic region, so this is where our fish are coming from, where depending on the temperature and the seasonality, you will have more or less estradiol. So estradiol was one of the hormones, the estrogen one I was talking to you about. So for example, in the Baltic Sea, you can have these kind of ranges of concentrations. But for example, when you go even to the Rhine area where you have near industrial areas, you can have really high concentrations of, for example, this estradiol hormone. And the reality is there's a lot of studies that have been done in guppies and other species of like zebrafish, but there's really not much, if anything, on our zygnatids, so we really don't know how these hormones are affecting them. And so this is why it was so important for us to really do this baseline kind of research that I showed you before, to get an idea of what are the hormones that these fish actually have, because the next chapter that I've been working on with my colleagues is really seeing, okay, with Zygnata stifla and nidophosophidium, when we add, for example, estradiol and 11-ketotestosterone and the mix to the water for several weeks, how is this affecting the fish? How is this affecting them just physically and morphologically? And we're looking then at gene expression, ELISAs, and the microbiome. So we're really wanting to get a big picture of how is this affecting the fish. And there's maybe a few results I can already show. This is really brief, and you let me know. So the main thing that you're looking at here, this was Zygnata stifla, and this is nidophosophidian. This is the gene expression analysis results. So the main thing I just want to give as a whole message is that the strongest effect, so you can see here at the top we have the males, and below we have the females. So sex is what was different. But what was really interesting is that we can see that the controls and the 11 kilo testosterone were actually really similar, but the estradiol was splitting off with the mix. And this is probably an indication that we know already that estradiol tends to strongly accumulate in water. And this is likely also what was happening with us. But there's still more work that we need to do. And we had a similar pattern in another tissue. Okay, and I'm done. But yeah, thank you everyone. That's pretty much just to say we have an effect. And thank you to all my colleagues and everyone.